Hello, it's Mr. Cho once again, and today we are going to be talking about the movement of substances, right, in plants, right, both the food like sucrose as well as water and mineral salts. Okay, so let's talk about this term called translocation, right? This is the transport of food that's mainly sucrose in the phloem tissues, and we will actually study this process using the translocation studies. All right, so we know that animals, right, we have a circulatory system, right, that's able to move blood around our body in order to transport substances. Like for example, our human, we have our heart, we have our arteries, we have our capillaries, our veins, etc. Right? All the blood vessels that move our blood around our body. But what about plants? Do they have blood? Right? Obviously, the answer is no, they don't have blood. So what system do they use to move substances around the body? Let's find out. Okay, so plants, they have a way of moving water and dissolved mineral substances around their bodies. And in animals we can measure their pulse, right? But in plants there's no heartbeat. So how do you actually study and prove, right, the movement of substances and food substance in the plant? Let's obviously start with a very simple experiment, which I'm sure everyone has seen before in primary school, maybe, right? So what you do is you simply take a living plant, right, like a celery plant, you cut the celery stem, and you put it into a glass of dyed water, blue or whatever color. And over a period of time, you will see that the xylem tissue will be stained blue, right? And if you look very, very closely, you will notice that it's actually only the xylem, it is not the phloem, right? So what conclusion can we draw from investigation is that, right, water will be transported through the xylem. Pretty simple experiment. So what about food, right? So we know that water is transported through the xylem. What about the food substances? So I don't know if you've ever seen this, but the bears sometimes they actually, they rub their bodies against the tree trunk, right, to scratch the itchy back or something. As they are doing so, sometimes they actually cause the rubbing, right, to rub off a complete ring of bark from the tree trunk. And this is called as girdling or ring barking. And this girdling can actually result in the death of the tree over time. Okay, so as you can see this example over here. Now, humans also practice girdling to remove trees in the forest, right? So actually, it's a very effective way, right? To I guess to sometimes you need to remove the trees, right, from certain parts of the forest. But how does the removal of a complete ring of bark result in the death of the tree? So evidence has shown that the girdling removes the phloem tissue that transports manufactured food substances from the leaves to the roots. Let's talk about this term, translocation. So translocation is the transport of manufactured food substances such as sugars and amino acids in the plant. Now, if you recall, in the previous part where I talked about the fate of glucose, right? We know that glucose can actually be transformed into sucrose, and this sucrose can actually be moved through the phloem, right? And so this is why it calls translocation. And amino acids, they also move through this process called translocation. So what evidence do we have? Okay, so the first evidence is called the ringing experiment. In this case, exactly what we saw here, a ring of bark with the phloem removed, you cut a ring of bark with the phloem is removed from the stem of a woody plant. So after some time, the stem immediately above the cut region swells up. As you can see over here, this is the region above the cut, it swells up. This is because the phloem is being removed and the food substances from the leaves, they cannot reach the stem below the cut region. Okay, why is that the case? Right, we know that the food actually moves through the phloem, but as it reaches here, right, it's empty space, right? And the food substance, it cannot jump, right? It cannot jump across, so it gets stuck in the phloem. Okay, and this will cause it to swell up. It actually can look quite drastic. It can actually be like really big, right? Like a few centimeters in its width. Okay, but what about water? Would water be able to move up from the roots towards the leaf? And the answer is yes, right? Because even though the ring of bark with phloem is removed, the xylem actually remains intact. So movement of water is still going to happen, and this will allow the plant to survive for maybe a few weeks, maybe a few months, depending on how durable the plant is. But eventually, right, if this is the only part, I mean, if it's like a big part of the tree trunk, then eventually, yes, it will cause the roots to die, right? Because the roots don't have enough food, and without energy, the roots are not able to, do their respiration, and then the whole plant starts to die. Okay, so we can try it. It's not a difficult experiment, but you do need to find some twigs, right? And to do this so you take two twigs, right? One with the bark removed, right? So you remove the phloem, the other one is intact, and eventually we'll find that um, A will die. Okay. Okay, so there are other ways to also prove translocation, and this one as way is using aphids. Okay, so aphids are insects that actually feed on plant juice. They have a long mouth part that's called a stylet, or also known as a proboscis, that is able to penetrate the leaf or the stem. So this aphid is able to insert its feeding stylet into the phloem. Okay, so it's just long enough to reach the phloem, but usually it's not long enough to reach into the xylem, which is usually deeper, right, in the inner ring of the stem. Now, what the scientists do is that they actually can anesthetize this aphid using carbon dioxide, which means that it numbs it, gives it it's a coma, right? Then animal rights, but whatever lah, it's an insect, right? The body is cut off, and only the feeding stylet is left remaining. So what happens is that it's like a bit like a straw now, right? It's a bit like a straw, and a liquid will exude from the cut. Exude means comes out, right, from the cut of the stylet, and you can analyze this liquid using pretty advanced tools. Obviously, it's a very small amount of liquid, so most likely you can't really use like things like bandit test or whatever. Okay, anyway, bandit test wouldn't work sucrose, but then you have to use like more advanced tools. So this will show that it contains sucrose and amino acids. If this stem is sectioned and examined under the microscope, you can see that the feeding stylet of the aphid is inserted right into the phloem sieve tubes. And this proves that translocation of sugars and amino acids occurs in the phloem. Okay, one more diagram over here. Right, this is a stem. Now remember, in the stem, we have the inner ring and the outer ring. Let's say that the outer ring contains the phloem, then in between that, there's a cambium, and the inner ring is the xylem, right? So usually plants in their stem, they try to protect their xylem, more important, right? Water more important, and food slightly less important, but not say, it's still important, but not as important. Okay, the third translocation study is called using isotopes. Now, an isotope is basically kind of like an element, right? But it contains usually contains extra neutrons, right? Extra neutrons. And so let's say for carbon, right? Normally carbon, it has, I think, 6 neutrons. Oh my gosh, my chemistry. Okay, yeah, 6 neutrons, or total of 12 lah, right? In the nucleus of the carbon, there should be 6 protons and 6 neutrons. Okay, but in this special carbon 14, right, it has an additional 2 neutrons. And what happens is that this carbon 14, right, it can undergo what it calls radioactive decay. So basically, this radioactive decay, right, can be detected using an X-ray photographic film. So what we do, or what scientists do, is that they provide this leaf, right, with this radioactive carbon, right? So this leaf is able to absorb the radioactive carbon that's provided to it in the jar, and it will be able to use the radioactive carbon to create its glucose, and that will create radioactive glucose. All right, so right, the sugars that form that radioactive glucose will contain this radioactive carbon, and then you can actually cut the stem and expose it to an X-ray photographic film. So what you can see is that eventually it is the phloem, right, the outer ring, which will show up on the X-ray film, and the xylem will not show up on the X-ray film. Okay, so these are the 3 tests, right, to prove that translocation occurs in the phloem. Okay, I'll leave you to do this yourself. Let's talk about the next thing. Okay, so now we know how the food substances, right, are moved through translocation from the leaf all the way down to the stem to the other parts of the plant. Translocation can also take place in the reverse direction. That means from the roots back up to the leaf. If let's say that there's no photosynthesis, food substances can move in both directions. Okay, but when we talk about water, we need to remember about the root hair cell, right? And we also need to talk about the movement of water between the plant cells using water potential. So when you water a plant, you pour water into the soil where the roots are, and usually you don't pour it directly onto the flower or onto the leaf, right? Because that will actually suffocate the flower or the leaf. So we know that water will enter the plant through the roots, but how does plant actually absorb water from the soil? So let's take a look at this picture over here. So if you zoom in all the way, we can see that this seedling is showing some root hairs. So each root hair has a narrow extension right over here, and this narrow extension, it pushes itself between the soil particles, and it comes into very, very close contact with the soil solution around it. So let's say that it's raining or the soil is wet, right? And so what's going to happen is that there's a thin film of liquid that's surrounding the soil particle, and that is actually a dilute solution of mineral salts, right? So this thin film of dilute solution mineral salts, what is going to happen? First, we know that water is able to enter into the root hair cells, right, by osmosis. So because the cell sap in the root hair cell is a relatively concentrated solution of sugars and salts, this cell sap over here has a lower water potential than the soil solution. The cell sap and the soil solution outside, these are separated by that partially permeable membrane in the root hair cell, and so water can enter into the root hair cell by osmosis. Now once the water enters into the first cell, let's say cell A, the entry of that water would then dilute the cell sap. This causes the cell sap of root hair cell A to have a higher water potential as compared to the next cell like cell B. So water will then pass again by osmosis from the root hair cell into the next cell, to B. And then this process repeats again, right, from B to C, because it's a continuous chain of events, right? So whenever there's a higher water potential in one cell, it will then move on, right, to the lower water potential, right, the cell with the lower water potential, and move, right, until eventually it reaches this red area, all right? And we see that this red area is actually our xylem, right, our xylem vessels. Now, xylem vessels are not cells per se, because they're actually dead. It's actually just a cell wall, a lignified cell wall. You can see this red is actually referring to a lignin. So once it has reached this xylem, it's able to be pulled up the stem by this process called transpiration, okay, which we'll talk more about soon. Okay, so basically water moves through the roots by osmosis, right, from a region of higher water potential in A all the way until the lowest water potential, which will be at the xylem. Okay. And what about mineral salts, right? If you just a quick one, right? What about mineral salts? Mineral salts will move into the cells by both a mixture of diffusion and active transport also. All right. Yeah, sorry, just repeating myself over here. Okay, so root hair cells absorb ions and mineral salts by active transport when the concentration of ions in the soil solution is lower than that in the root hair cell. So you have to absorb it against the concentration gradient by using active transport, and you actually have to use energy for cellular respiration in the root hair cell. So actually in the root hair cells, they do have a bit more mitochondria than normal, right? A normal cell to give this energy for active transport. But mineral salts can also enter by diffusion when the concentration of the ion is in the soil is higher than the root hair cell. So how is the root hair cell adapted to its function of absorption? There are a few things. Firstly, root hair cell has the root hair is a long and narrow extension of the root hair cell. This increases the surface area to volume ratio, which in turn increases the rate of absorption of water and mineral salts by the root hair cell. Okay, so it's an important point over here. Always remember, whenever you talk about the structure, you also need to talk about the function, right? The structure is a long and narrow extension. The function is to increase the surface area to volume ratio, which will then increase the rate of absorption of water by osmosis and mineral salt by active transport and diffusion. Now, the cell membrane actually prevents the cell sap from leaking out. So the cell sap will contain sugar, amino acids, and salts, and this will cause a lower water potential in the cell sap as compared to the soil solution. So water can enter by osmosis. We have cells containing many mitochondria. Aerial respiration in the mitochondria can release energy for active transport of ions in the cell. Okay. All right. So you can do this yourself. Let's talk about the next parts. How does water move up a plant? Okay, so we're going to learn about this very important term called transpiration. Okay, we're going to learn about how water can be pulled up from the xylem vessels towards the leaf by transpiration pull, and we're going to learn about air movement, temperature, humidity, light intensity, how it affects transpiration rate, as well as how wilting occurs. Okay, so there's this tree in Singapore called the Seraya tree, and this Seraya tree is like I think it's like 50 meters tall, maybe about as tall as like a 30-story HDB. So you'll be wondering, like, how does water get all the way from the roots, all the way carrying it up 30 stories, right, all the way to the leaves? Right, actually, there's this force that pulls. Remember, a force is something that can pull or push. So there's a force here that's called transpiration pull. And this transpiration pull, the force, is created during a process called transpiration. So what is transpiration? Now, if you have ever been to a forest and you feel like, how come the air feels so humid? Right? One of the reasons why is because the plants are constantly transpiring around you and they're releasing water vapor out from the leaves. So the green plants, they absorb water from the soil, but not all of the water that's absorbed will actually be used up by the plant. Okay, so the plant can use water for a few things. Firstly, it helps to keep the plant turgid. Secondly, they use the water for photosynthesis, right? But they don't use all of it. So the excess water actually can be removed when it evaporates from the aerial parts of the plant. Aerial referring to the stem and to the leaves, the upper part, the part that's not above, above the soil. So transpiration is the loss of water vapor from the aerial parts of plant, mainly through the stomata of the leaves. All right, so take note, this is a definition. Please memorize this definition about transpiration. So how can we actually test for transpiration? There are a few ways we can do that. So one way is by putting this plant inside there's a plastic bag here, right, to prevent any of the gases from the soil from coming out. And what can we do? All right, so we can do a few things to it. Let me see. Okay, let's move on. Okay, so let's look at this one. This one's more meaningful. Okay, so let's say that we have two twigs of about the same size from the same plant. Okay, so in the first twig, we cut the end of each twig underwater to prevent air from entering the xylem vessels. Okay, so this is the cut end. And any air that's trapped in the xylem vessels will interfere with the absorption of water by the twig. Okay, so we try to make sure that water can move through the twig properly. So you place each twig into a beaker of water and add a little bit of oil, right, to prevent evaporation from the upper surface of the water. You strip off all the leaves of one twig and put petroleum jelly on the end of the leaf stalks, right? So over here, you put petroleum jelly. It's just a kind of like jelly that prevents evaporation. Now, you also put approximately the same amount of petroleum jelly on the stem of the leafy twig, right? So just the stem, not the leaves. Now you place each beaker under light, okay, so that they can perform photosynthesis also. Okay, so what will happen is that over some time, we will notice that for, let's call this A, for A, the water level will actually drop. Why? Because water will be moving through the stem, the xylem in the stem, moving up, right, into the leaves, all these leaves, and it will actually evaporate out from the stomata and into the let's call this the atmospheric air around it. Okay, so the water will drop over here. But for B, the control will just remain the same, right? The water level will not drop. Okay, so you can actually measure you can measure the rate. right? How much water drops, we can measure in terms of height, we measure in terms of the volume of water loss per hour, yeah, you can, there's a lot of ways you can do it, okay? Okay, so this experiment, if you want to compare the rate of transpiration on the upper and lower surface of the leaf, you can actually use this thing called cobalt chloride paper. So cobalt chloride paper is blue when it's dry, and it turns pink or somewhat whitish pink when it comes into contact with water vapor. Okay, so what we do is that this, you just see this here, there's a leaf and there's dry cobalt paper on the top and dry cobalt paper at the bottom also. So what we would expect to see is that because there's more stomata at the bottom of the leaf, right, more water vapor will actually be removed, will transpire out from the bottom, right, and so the bottom cobalt paper will actually turn pinkish white sooner than the top cobalt paper. Okay, so that means that will show that the rate of transpiration at the lower surface of the leaf is higher as compared to the upper surface. Okay, so again, same thing. Okay, if we say do one experiment with the petroleum jelly, if you have leaf A, we cover the upper surface with a layer of petroleum jelly. Leaf B, you cover the lower surface, and leaf C, you cover both surface. Which do you think will actually lose the most water? The answer is actually A, right? Leaf A, because the lower surface is exposed, right? So you lose a lot of water. For leaf B, the lower surface is covered, the upper surface is exposed. You will lose some water, okay, but not a lot. And for leaf C, you will probably not lose much water because both surfaces are covered. So top and bottom both cannot do transpiration. Okay. Okay, so let's talk about the process of transpiration. Okay, now this one is quite, I'll say, a little bit of physics involved lah. Okay, so let's try to understand it. So transpiration, right? The evaporation of water from the leaves removes water from the xylem vessels, and this results in a suction force which pulls water up the xylem vessels. This suction force due to transpiration is known as transpiration pull. It is the main force in drawing water and mineral salts up the plant. Okay, now transpiration pull is the main force, but there's actually two other forces, right, that actually help it, called root pressure and capillary action. Okay, I'll talk just about capillary action. Basic capillary action is a very simple process, right? When there's a very narrow area, water tends to move up. It's just like, for example, you put your straw inside a very narrow straw, you actually notice that the water will rise up slightly inside the straw. Okay, and that is called capillary action. So, but for transpiration pull, right, this is the main force, right, the main suction force that pulls water up. How does it work? We know that water is absorbed from the soils into the root hair, right? We just talked about it just now. And now the water will move, right, from high water potential, low water, from cell to cell to cell to cell, right, until eventually it reaches the xylem, and then it will move up the xylem because of the transpiration pull. How this happens is that this water evaporates and escapes as water vapor through the stomata and the leaves. So as the water leaves the stomata, it actually creates that pull, right? That pulling force, that transpiration pull. And because water, right, if you ever notice, right, water, it tends to move together. Like let's say you have a cup of water, and when you touch the water, you notice that water sticks together, right? I don't know how to describe, but there's this like stickiness of water that can be observed. There's actually called, there's a name for it lah, right? The surface tension and all that kind of thing. Basically, water pulls each other. You can think of it like the water, they're holding hands, right? They're holding hands, and as one molecule of water evaporates, it pulls the next molecule of water into the leaf, and it's a continuous chain. Right, so this whole process happens over and over and over. Water vapor evaporates, that's called transpiration. That creates a transpiration pull, which pulls more water up the xylem vessels into the leaves. As long as there's water in the soil, this process can keep on happening and keep on happening, and this is how the plant survives for so long, right? Okay, let's look at how in more detail how this happens. So this figure shows the movement of water through the leaf during transpiration. right? Water evaporates from the surfaces of the mesophyll cells into the air spaces and then diffuses out of the cell through the stomata as water vapor. Okay, so this is how it happens, right? Water moves up the xylem, right, in this here. And once it reaches to the cells, right, it will have, again, it will move by water move by osmosis, right, towards all these different cells, right? But what we care more is mainly over here. So we care about these cells that are near to the intracellular air spaces. So that's it. Yeah. Okay. So what happens is that let's go step by step lah. Let's say that the water moves here, and you see this blue line, right? This blue line is actually a thin film of moisture that surrounds the mesophyll cells. So water would evaporate from this thin film and form water vapor in these intercellular air spaces. I think just now I said intra, right? So I meant inter, right? Intercellular air spaces. Now, because there's a high concentration of water vapor, okay, now remember when we talk about water vapor, it's going to go through the process of diffusion, no longer osmosis, right? A bit confusing, but water vapor, diffusion. Liquid water molecules, osmosis. Okay, anyway, step 2. Water vapor diffuses through the stomata towards the drier air that's found outside the leaf. And this diffusion is known as transpiration. Now, because the water evaporates from the thin film of moisture, more water will then move out from the mesophyll cells to replenish this thin film of moisture, right, as we see in step 3. So the mesophyll cells will draw water from the cell deeper inside the leaf by osmosis, right? Because again, this is the chain, right? It's a chain reaction, right? This one has low water potential, so the cell if behind it has higher water potential, then it keeps on moving. Okay, and then the cell that's deeper inside the leaf will then draw water from the xylem by osmosis. And this actually creates the suction force, which pulls the entire column of water over here up the xylem vessel. This force is called transpiration pull, right? And so this process happens again and again and again and again until the plant eventually dies. Now, because transpiration occurs through the stomata, it is also linked to gas exchange between the plant and the environment. Okay, what I mean by link? That means that when gas exchange is happening, it affects the rate of transpiration. So for example, in daylight, the stomata actually opens wider to allow carbon dioxide to diffuse into the leaf for photosynthesis. So oxygen and water vapor are more concentrated in the intercellular air spaces, so they will diffuse out of the leaf through the stomata. So this actually creates a very interesting dynamic for the plant. What I mean by that is that sometimes the plant is smart. I'm not saying smart, I mean the plant is pretty smart. What this means is that if the sun is very, very sunny, it's very, very hot, by right it should be good for photosynthesis. But by left, right, it causes too much evaporation, too much transpiration, causes the plant to lose too much water, and then that's not good for the plant. So the plant has to balance its demands, right? It wants to have carbon dioxide to enter its intercellular air spaces, but it also doesn't want to lose too much water at the same time. So it needs to balance the size of the stoma between these two factors. Okay, so what is the importance of transpiration? We know that excessive transpiration is actually harmful for the plant, right? This is what causes wilting, then the plant don't have enough water. But transpiration actually has a lot of uses. In fact, transpiration is a key part of the plant, because transpiration pulls water and mineral salts from the roots to the stem and the leaf. Without transpiration, the leaves cannot survive. And the evaporation of water from the surfaces of the cells of the leaf cools down the plant, preventing it from being scorched by the hot sun. And so actually it will actually remove all this heat that's coming from the sun. And water that is transported to the leaves can also be used during photosynthesis. It can also be used to keep the cells turgid, and it can also be used to replace water that is lost by the cells, right? For example, to maintain that thin film of moisture. And these turgid cells will help to keep the leaf, turgid means like it's very plump, right? The cells can keep the leaf spread out widely in order for the plant to trap enough light for photosynthesis. Let's look at this figure to measure the rate of transpiration. Okay, so the first method is using a spring balance. Right, so a spring balance, it measures the weights, right? Okay, the weights of this whole setup. So let's actually weight mass. Okay, let's just say mass lah. Okay, mass. Right, the mass of this setup. Okay, what happens is that you can just put a leafy shoot, right, that is able to absorb water through transpiration, okay, and give it some time, maybe 5 hours. And you also put a layer of oil here to prevent any evaporation from the upper surface. What we do is that, let's say the mass of the tube with the shoot in the initial is A grams. After 5 hours, right, the mass of the shoot after 5 hours is B grams. So what we do is we take A, initial mass of the tube, minus B, the final mass of the tube after 5 hours, divided by 5 hours. And we can get the amount of grams lost per hour. This is the rate of transpiration. Okay, now remember, when you're writing in the exam, do write down how you can measure the rate of transpiration. Okay, so you need to write this whole more or less this whole long story out. Okay, it's quite long, but yeah. Now give your measurement of mass and rate to two significant figures. Okay, in this case, just follow the exam instructions. If they say 2, follow. If they say 3, follow. Okay, this experiment, sometimes if you don't have a spring balance, what you can do is you just measure the height lor. You can also go by height, but that's not the best way to do it. All right, so preferably there's a spring balance provided. Okay, there's also another way, which is using this thing called okay, this one I don't want to talk too much about it lah. It's very similar to the previous one, but the second way is actually using this thing called potometer. Okay, I don't know why they have to put these guys here, but anyway, it's also quite a simple concept. Let me see if I can find the example here. Okay, so how we can use this potometer, and why we use the potometer is because actually it's quite difficult to measure the rate of transpiration directly, right? Because we also know that water can be used up in photosynthesis. So it's not just transpiration happening, but photosynthesis is also happening. So we can measure the rate of water absorption using a potometer. And so this is an example of potometer. Okay, you can see that there's a plant here that's inside this tube. This is a beaker of water. It's a bit hard to see, but there usually is a bubble, right, or meniscus somewhere around the line. Right, you can actually measure the movement of the bubble, right, from the beginning point over here until wherever it moves, right, however fast it moves. Okay, so there are a few things to know about potometer, but that's not very important. I doubt you will have to, I mean, if you really have to do it, then just follow the instructions. Okay, basically try to make sure that there's no bubbles or anything that are unnecessary bubbles. Okay, so a potometer can be used to determine the effect of different environmental conditions on the rate of transpiration. For example, you can measure the effect of temperature on the rate of transpiration. Okay, so you can use a room with an air conditioner where you can actually control the room of the temperature of the room. Maybe you can set 5 different temperatures, right? 30, 25, 20, 15, 10, something like that. Okay, I don't know if the aircon can go that cold, but I just imagine. Okay, you can also investigate the effect of wind speed, which we'll talk about later also, because all these factors are these 3 factors: temperature, wind speed, line of sight. These are all factors that can affect the rate of transpiration. Okay, so how do we use a potometer? So firstly, we have a chute, and we put it through this cork over here, right, into this what I call as there's just the water over here. Now make sure that this part is sealed very well. So you can put some petroleum jelly in between these layers to seal it very perfectly and prevent any evaporation. Okay, so airtight. Then you open the tap. Okay, but it's all just procedure. But what's important is over here. Okay, the key concept is that when transpiration occurs over here in the leaves of this shoot, it will cause the water to move from point B at the end of the water column towards point A, right, through this graduated capillary tube. The word graduated simply means that there are lines on it. So what you can do is you can measure. Let's say that this is 0 distance, this is 1 cm, 2 cm. You can measure the distance that's moved by the water, right, the meniscus, and over a period of time, let's say 5 hours, right? So if it moves really fast, then it means the transpiration is occurring really fast. If it moves very slowly, then transpiration is occurring very slowly. Okay, so similar story, we can take the volume of water from B to A. We can say that this is A volume, right? And we say that the time taken is B minutes. We can measure the rate of transpiration by taking the volume of water that's lost divided by the time taken in minutes to get the rate of transpiration to two significant figures. All right. So what factors can affect the rate of transpiration? So these are the three factors over here: air movement, temperature of air, and light. Okay, there's one more factor that someone is missing. Don't see here. It's humidity. So how I remember it is I remember the word HALT, HALT. Humidity, air movement. Oh, it's over here. Humidity, air movement, light, and temperature. HALT, HALT. Okay, so let's talk about air movement first. So in still air, water vapor will diffuse out of the leaves and accumulate around the stomata. Okay, so imagine that the leaf, outside the leaf immediately, right, there's a bunch of water vapor. Now this accumulation of water vapor will cause the concentration gradient of water outside to inside and outside to run less steep. So now the amount of water vapor outside is quite similar to the amount of water vapor that's inside the leaf. And so the rate of transpiration is lower. But if wind is present, the water vapor that diffuses out of the leaf will be blown away and it does not accumulate. Thus, the concentration gradient of water vapor inside and outside of the leaf will become steeper and the rate of transpiration will become higher. So the stronger the wind, the higher the rate of transpiration. You can think a bit like, sometimes people, they're going to stand outside the door. Then you want to exit from the door, you cannot move. But if the security guard come and blow all these people away, chase them away, then you can walk out of the door very easily, right? Something like that. Okay, the temperature of the air. So we know that temperature, right, hot temperature, high temperature will increase the rate of evaporation of water. So the higher the temperature, the faster the rate of evaporation of water from the cell surfaces, and therefore the rate of transpiration will be higher at high temperature. So nothing much to elaborate. Okay, light, it affects the size of the stomata of the leaf, right? Because when there's more light, more photosynthesis, and then the gut cells will open wider. And when the gut cells open wider, the stomata also opens wider, right? This will affect the rate of transpiration. So in the presence of light, stomata opens wider, increases the rate of transpiration, but in darkness, the stomata will close and less water will be lost from the leaf. Now remember, stomata doesn't close like 100% of the time. Not 100, like 100% close. It's like at most also like 99% close. Okay, so there's always going to be some movement of water, but just lesser lah. Okay, but cannot be like perfectly no water at all. Last one, humidity. Okay, so humidity of the air refers. Some people get a bit confused about the word humidity. Humidity refers to how wet the air is, basically, right? So high humidity is like in Singapore, right? The whole air around us feels very wet all the time. We feel very sticky and sweaty. But in some countries like very dry, like Australia, right? It's low humidity because the air is very dry. So normally the intercellular air spaces of the leaf are saturated with water vapor. So there's a concentration, water vapor concentration gradient between the leaf and the surrounding air. When the leaf outside the air is drier or less humid, the concentration gradient of water vapor will become very steep. Thus, the rate of transpiration will be faster. Increasing the humidity of the air will make the water vapor concentration gradient between the leaf and the surrounding air less steep. Thus, the rate of transpiration will decrease. Okay, so when it's outside is too humid, the plant is not able to transpire properly. So actually, this can be good and bad, right? For example, some plants, they don't want to lose water, so they like having a humid environment. Some plants, they want to lose water, so they need to have a drier environment. But let's say this plant, right, this is called a marram grass, right? This plant, they actually live in very dry conditions, so they don't want to lose so much water. And they have this special structure that help them to control the transpiration rate. They actually have this sunken stomata, right? And these grooves, all these little grooves over here, they have hair. This hair help to trap the water that's diffusing out of the stomata. And then this will actually increase that humidity in this area, and overall that will reduce the rate of transpiration so that they don't lose so much water, because it's a dry environment for them. Okay. Okay, we're coming to the end of this chapter. One last thing, let's talk about wilting. So we know that in each of the plant cells, right, there's a cell wall, there's a cell, the large central vacuole. So in the vacuole, you have the cell sap. Inside the cell sap, you also need water. So when there's enough water in the cell sap, that's why it's called as having turgid. What does it mean? Right? There's this pressure, right? This thing called the turgor pressure. They are inside the leaf mesophyll cells, and they help to support the leaf and keep it firm. Basically, that because the cell sap is very full, right? The vacuole is very full with cell sap, right? It pushes against the cell wall and causes the plant to be firm. Now, this will actually enable the plant to spread out widely, the leaf, right? Spread the leaf widely and absorb light, more light for photosynthesis. In strong light, when the rate of transpiration exceeds the rate of absorption of water by the roots, the cells start to lose their turgor, right? So now the plant becomes flaccid, right? Flaccid is the opposite of turgid, and the plant wilts. Wilting also can occur in some of the plants with soft stems, and then the whole plant will actually like kind of topple over. Okay, so there's advantage of wilting. Okay, it's not actually a bad thing. So when the plant wilts, right, there's a reduced leaf surface area, and that will decrease the rate of transpiration. Remember that water is very important for the plant also, right? And so it cannot lose too much water, if not you'll completely die. So this is a strategy, right, for it to reduce the surface area that's exposed to light. And when it reduces the surface area, right, it reduces the amount of exposure of stomata to the atmosphere and reduce the rate of water loss through the stomata. Right, this excessive loss of water causes the gut cells to become flaccid and the stomata to close. So overall, rate of transpiration is reduced. Right, so this helps to save the water that's in the plant. But of course, the disadvantage, if the stomata are closed, the amount of carbon dioxide that's entering the leaf will be reduced. And then carbon dioxide becomes a limiting factor, and the rate of photosynthesis will become decreased. So the folding of the leaf also reduces the surface area that's exposed to light and also reduces the rate of photosynthesis. So again, as I told you, it's a balance. Right? Plants need to balance between these two states. They want to do photosynthesis, but they don't want to lose too much water. So how? They have to use both modes in a strategic way. When it's too hot, they can wilt. When it's not too hot, right, it's nice temperature, everything, early in the morning with enough sunlight, they can become more turgid, and then they can absorb more sunlight. Okay. All right. So I think that is it for this whole chapter. Right. Do look through your textbook, where you can try some of these questions here and there. Answer keys actually at the back of your textbook. Okay. With that, yeah. Wish you all the best in your studies, and I will see you in school. Bye bye.